Today's CSEB webinar is entitled Big Questions in Cancer Epidemiology, Confounding Our Future. This webinar is presented by Dr. Margaret Spitz of the Baylor College of Medicine in Houston, Texas. Dr. Margaret Spitz joined the faculty of MD Anderson Cancer Center in 1981 and was named founding chair of the Department of Epidemiology in 1995 until stepping down in 2008. She joined Baylor College of Medicine in 2009 to provide strategic direction in growing their population sciences program. Dr. Spitz has a long-standing interest in genetic susceptibility to lung cancer. She has developed a lung cancer risk prediction model, has participated in lung cancer GWAS, and is a founding member of the International Lung Cancer Consortium. She is principal investigator of a recently funded R25 in pediatric cancer epidemiology. Nationally, Dr. Spitz served as a co-chair of the NCI's Lung Cancer Progress Review Group and recently completed service on the board of scientific advisors of the NCI. She sits on the external scientific advisory committees of several major cancer centers. She has served on the National Institutes of Health Studies section and is past president of the American Society of Preventive Oncology.
Thank you very much for this opportunity to present what I think are some of the provocative questions that cancer epidemiologists are going to be dealing with in the next few years and actually already are dealing with. So I'll begin with my take-home messages and proceed from there. The first take-home message is this is an exciting time to be a cancer epidemiologist, I believe that epidemiology has gone mainstream. Certainly in cancer centers this is true. When I started my epidemiology research, we were considered almost second-class citizens, but now with the emphasis on team science and translational research and clinical application, it is clear that epidemiology has gone mainstream. And finally, I hope to convince you all that epidemiology can provide the framework for the large integrative studies that are now being conducted to look at cancer risk and cancer outcome. And the questions that we're asking now remain the same as the questions we were asking when I first started and when I conducted classical epidemiologic studies. Why did this patient get this disease at this time? Could this have been prevented? And could this cancer have been predicted? And it is this issue of cancer risk prediction which is a particularly difficult problem to address and to try and sort out. As Niels Bohr, actually the physicist, said, predictions are risky, especially about the future. And although it's also been attributed to Yogi Berra and even to Dan Quayle, as strange as this sounds, it is extremely difficult to predict the future. And this cartoon will illustrate why. Here is Britain's oldest smoker who died at the age of 103. She started smoking, in fact, when she was 7 years old, when the First World War broke out, because she said she needed to calm her nerves. She actually outlived 2 husbands, her son, and 10 stepchildren. She only stopped smoking about 3 months before she died, because due to failing eyesight she could no longer see the end of the lighted match. Certainly one would not have predicted that a smoker who smoked for 103 years, or for actually just -7, would have lived this long and appear to be so healthy. So this just illustrates the difficulties in predicting outcome. So when I started in the 1980s, we were doing classical epidemiology. The tools we had were pen and paper, and we did detailed questionnaires, and we associated risk factors such as smoking, diet, family history, occupational exposures, and so on with cancer risk. And classical epidemiology has made major contributions to understanding risk factors for cancer, and one must never underestimate the importance of a good questionnaire in any type of study. Then in the mid-1980s, the concept of molecular epidemiology emerged, and this approach was to incorporate molecular and biochemical markers together with questionnaire information to try and understand the mechanisms of carcinogenesis and events throughout the continuum between lifestyle behavior, exposure, and cancer risk. And the markers that we were looking at were markers of susceptibility, markers of exposure, and markers of effect. To some extent, the long-term goal of such an approach was to develop a new prevention paradigm in the same way that at the same time personalized medicine was being developed, in which a particular molecular genetic aberration was targeted for targeted therapy. In the same way, we hoped to investigate different risk profiles and target prevention programs depending upon the risk profile of the individual. And as you know, there have been major successes in targeted, personalized treatment of cancer for a variety of new cancer drugs, and we wanted to have the same approach for cancer prevention, so that if an individual had an average risk, we would recommend behavioral change such as stopping smoking, using sunscreen, and so on. Slightly higher risk individuals, we might recommend a screening program that would not perhaps be appropriate for the general population. For example, women with a BRCA1 mutation should be screened with MRI every year or every 6 months, and this would not be appropriate for an average risk woman. The highest risk individuals might be recommended to have prophylactic surgery, and obviously this is not something that one undertakes lightly, but perhaps a woman who's BRCA1 positive, who's had her children, and might be recommended to have oophorectomies. Then, of course, I didn't put in here, but we have the whole area of chemo prevention for high-risk individuals. So the hope was that by carefully characterizing the risk profile of individuals, we would have targeted prevention programs. I became chairman of the Department of Epidemiology in 1995, and I was actually very disconcerted when a perspective was written in Science by Gary Taubes, whose thesis was that epidemiology had exhausted its potential. And he quoted from a variety of very well-known epidemiologists, one of whom said that epidemiology is generating conflicting results, which confuses the public and disoriented policymakers. It was claimed that epidemiology was forever sounding false alarms, that we have found all that we can, that methods of epidemiology are too crude to detect anything but the largest effects. And my favorite of all was that the future lies not in the lab, not in epidemiology, but in the laboratory, as if the laboratory was not part of epidemiology. To make matters worse, a leading oncologist at the time said that epidemiologic research was phenomenologic. Well, I actually hadn't heard that term, and I briefly wondered whether he meant phenomenal, but I detected the sort of sarcastic tone, and I spent many years after that trying to prove that epidemiologic research was not phenomenologic, but could provide a framework for large-scale integrative studies. And I was also a little bit mollified by an editorial which was written by Trikopoulos in the British Medical Journal the following year, in which he compared epidemiology to democracy. He said that both were not perfect approaches. They had their own inherent problems and weaknesses, but both represented the best available approach to achieve their respective objectives. And he pointed out that epidemiology was likely to continue and to flourish, but one must just be cognizant of the inherent biases and problems inherent in epidemiologic research. And I felt much better, but he did spoil it a little bit by saying that few epidemiologists are equipped to undertake the task. And if truth be told, we then had the era of candidate gene studies, and Michael Toon this year called this the lost decade of candidate gene studies. We were limited by relatively low throughput of existing platforms, so that our candidate gene studies were small and they lacked power. So we were very happy to publish a candidate gene study maybe of 300 cases and 300 controls, and looking at 5 gene variants or single nucleotide polymorphisms in a gene, which was almost invariably published if it was positive. But the problem was that very few of these associations were replicated, and even fewer of the gene-environment interactions that we demonstrated were reproduced. Part of the problem was, as Eric Lander has said, we were very poor guessers and we failed to pick good candidates to explore. And one of the problems was that we did not specifically focus on functional gene variations. So in fact, there was some problems with epidemiologic research. And another issue was there were existing dichotomies in research efforts driven by diverse disease-specific emphases and interests. For example, some of us were focusing on the environment, others on genes. Some were focusing on risk, others on outcome. Were we looking at somatic variants or germline variants? Were we focused on genetic factors or epigenetic factors? And these dichotomies did little to advance in-depth understanding, but in fact tended to obscure the complex biologic reality that needs the full participation of multidisciplinary teams of scientists if we are going to unravel these issues. So it was clear that we needed a new integrative approach that would coalesce these diverse domains and provide a cohesive umbrella. In addition, we were now transitioning to an era where we needed to apply advanced technologies which were providing substantially higher order of magnitude of data than we had when we were doing candidate gene approaches. And these technologies included high throughput platforms for genotyping and sequencing, all these different omics platforms, novel imaging opportunities, and also advanced statistical and bioinformatic tools. And these new technologies were helping us to much more carefully characterize exposure and outcome, and to give us greater opportunities to dissect the molecular basis of carcinogenesis. And it was clear that these new technologies were changing the landscape of epidemiologic research. And we also began to recognize that this approach required very thoughtful and deliberate collaboration across multiple disciplines, and also provided us with exciting opportunities to embark on large-scale collaborative studies that had the requisite tissue, biospecimens, and data that we needed for this new era of integrative approaches. And I would argue that epidemiology, as I've said before, provided the framework for this approach. It is clear that there is a bidirectional relationship between tools and epidemiology. The tools provided the data deluge into which epidemiologists could delve, and on the other hand, epidemiology has the ability to inform, test, and validate these technologies for wide-scale application. In addition, we know that these new integrative studies in which we merge the boundaries of molecular epidemiology with a variety of other disciplines that I'll go into in a moment, these large studies, the absolute cost is greater, but on the other hand, the marginal increase in cost per unit information gained is much less, and the scientific payoff is much greater. So here is our schematic of this new integrative approach where we are merging the boundaries of molecular epidemiology with genetics, basic biology, molecular pathology, clinical oncology, and biobehavioral science, as well, of course, all the types of systems biologies and systems genetics approaches that are now being developed. So in the old days, as I've said, we studied germline changes, largely SNPs and epigenetic effects and serum markers, and correlated them with risk. Now, in the new integrative era, we can do high throughput exome, whole genome sequencing, and we can explore the whole epigenome, both of the somatic and of the germline genome. And we're also recognizing that gene discovery and exploration of molecular underpinnings of cancer risk move seamlessly between studies of the somatic genome and epigenome, as well as genetic and molecular epidemiology, thereby informing and enriching both disciplines. And in addition, we are now able to study intermediate phenotypes using all these new omics such as metabolomics, transcriptomics, lipidomics, and so on. And we also recognize that not only would we study on risk, but we could also study prognostic and predictive markers of outcome. We could study markers of risk of second cancers. We could study risks of toxicities and so on. And this was a much more integrative approach where we were working with a multitude of different disciplines. And we could use the same platforms as we used in classical epidemiologic studies. But even though we are faced with the excitement of this new types of technologies, epidemiologic principles, concepts, and methods should never be abandoned. We need to have pristine data collection, pristine study design, and the most careful attention to analysis and biases. And one new approach is the so-called concept of extreme phenotypes. We recognize now that subjects at one or both ends of the extreme of any type of phenotype of interest are likely to be enriched for the traits, the variants that we're interested in studying, and that therefore if we use extreme phenotype subjects, we will be designing smarter, smaller, and more powerful studies. And finally, I wanted to add this little component. We are also recognizing now that we can use genetic factors to study behavioral lifestyle choices which place people at increased risk for cancer. And these behaviors include smoking, alcohol use, obesity, and physical inactivity. And this is a relatively new area of science which has been variously called behavioral genetics and biobehavioral research. And although we think of this as a new science, in fact, it's not new at all. Sir Francis Galton was a pioneer in behavioral genetics, and he exhorted us to study twins because he said we can distinguish between the effects of tendencies received at birth, nature, versus those imposed by the special circumstances of their afterlives, by which he means nurture. And we actually think of this as genes and environments and gene-environment interactions. We now also recognize that individuals live in societies. They influence and are in turn influenced by their families, their social networks, their communities, and the society at large. So when we talk about the environment in epidemiologic research, we are thinking not only the physical and the chemical environment, but also the psychosocial and the cultural environment in which individuals live. And this is a very important point to continually remember. So in 2010, I was invited to the first Big Questions in Cancer Research meeting, which was organized by the NCI. And I was the only epidemiologist there, so I felt that I had to represent the entire field and not just my own narrow area of interest. Originally, it was called Big Questions. We suggested understudied, neglected paradoxes, and we finally selected on Provocative Questions. And Dr. Varmus gave a very great overview of his concept of these provocative questions. And they need to address important problems and paradoxes in cancer research that had not been sufficiently investigated and studied. And these problems could be older observations that had been laying dormant for a long time, or they could be more recent perplexing findings. But the important things were that they should be non-obvious mechanisms, and that we needed to think outside the box and explore new ways in which we could understand these neglected observations and find out how they were related to cancer risk or to cancer, and to develop new and exciting cancer therapies. And as you probably know, there was a first RFA which had 24 provocative questions with 2 RFAs, and I think the first round have been funded and a new RFA has been issued. So I actually had 3 questions, only 2 of which were accepted, but that's pretty good going. And the first question was, how do we characterize the biologic basis of cancer susceptibility? I've added lung for my own interest, but I had been broader than that in the submitted question. What molecular mechanisms explain the association between obesity and cancer risk? And what are the relationships between the human microbiome and obesity? And I'm going to try and convince you that we can apply this concept of integrative epidemiology to answer these questions. Now, it's interesting that the questions were related to tobacco and diet and obesity, poor diet and physical inactivity, because this paper in JAMA fairly recently showed that both in the 1990s and in the 2000s, these were the 2 leading causes of death in the United States, accounting for about 1/3 of all deaths in the United States. And you can see here slight decrease in the percent attributed to tobacco, and certainly an increase in the percent attributed to poor diet and physical inactivity. And these are both modifiable risk factors, and that's why it's very important to explore these further. So question 1 is related to the hereditary component of lung cancer. We know that genome-wide association studies in which we explore genetic variants across the entire genome have provided us with quite a lot of information on the genetic basis of lung cancer and a variety of other cancers as well. However, the gap between discovery and clinical application is still very large, and these GWAS have explained very little about cancer heritability, and this has led to the hotly debated topic of missing heritability of cancer, and there are many publications on this that you can look at in the literature. Lung cancer is also different from most of the other cancers that underwent GWAS because we know the cause for 85% of all lung cancers, and that is cigarette smoking. So for lung cancer, we have to consider that we need to study the genes for nicotine dependence, which I'll discuss briefly, as well as the genes directly associated with lung carcinogenesis. And finally, the other important reason why this is so important is we need to identify high-risk smokers. And I'll explain this to you why we need to do so. We know that 85% of all lung cancers occurs in ever smokers. And if you were to draw the population response to long-term smoking in terms of lung cancer risk, you might see a curve like this. However, that would be the lifetime risk. But this curve does not show the highly exaggerated curve for susceptible smokers who develop lung cancer earlier and with lower levels of exposure. And there is a small group like the lady I showed you in the beginning who are relatively resistant to the adverse effects of cigarette smoking. But we do know that only a fraction of cigarette smokers will develop lung cancer. In fact, it's been estimated that the lifetime probability of lung cancer in the United States is about 1 in 13 for men and 1 in 17 for women. If you look only in ever smokers, it's about 1 in 6.5 for men and 1 in 10 for women. And I did a quick back of the envelope calculation, and there are about 45 million current smokers and more than that, 49 million former smokers in the United States. How do we identify that fraction of smokers most likely to get lung cancer, and why is this important? Well, this is important because the National Lung Screening Trial in the United States recently showed a 20% reduction in mortality in individuals who received CT screening compared with regular chest x-rays. Clearly we can't screen all these current and former smokers. We need to identify the highest risk subgroup, and it's those who we would enroll in these expensive screening programs. So you know, to identify genetic factors for lung cancer, there are two approaches. If you have for very high risk alleles, in other words with high penetrance but very, very rare in the population, the best approach is to do family studies with multiple cases, and you do linkage analysis. But what we are referring to here are the so-called low risk alleles, which are common in the population but associated with low risk, and here you have to do large population-based association studies, and that is what we've been focusing on. Now, first of all, let's get back to the concept of nicotine dependence. This cartoon slide just illustrates the power of nicotine dependence. Here is a bride smoking through a hole in her veil, and here is a surgeon during the SARS epidemic. This surgeon happens to be from Toronto, nothing against Canadians, of course. He was very concerned about his risk of SARS, but he didn't seem to be concerned about his risks either of chronic obstructive pulmonary disease or of lung cancer. But we know now through a variety of twin family-based studies that there is a genetic role in nicotine dependence, and there is a higher concordance rate of smoking or not smoking in monozygotic compared with dizygotic twins, and there have been fascinating studies showing the concordance rates of smoking or not smoking in twins who are raised apart. And as a result of these excellent family-based studies, we now estimate that there is a 60% heritability rate for the initiation of smoking and about a 70% for the maintenance of dependent smoking. And this has actually revolutionized the way we think about smoking. We now know that smoking is a complex trait, and it has gene-gene and gene-environment interactions, and there is also considerable phenotypic and genotypic heterogeneity, exactly as you would describe a particular cancer. So in order to understand something about the genetics of nicotine dependence, we need to know something about nicotine's pharmacokinetics, in other words, its metabolic capacity, as well as nicotine pharmacodynamics, that is central nervous system neurotransmitter functionality. And nicotine pharmacokinetics is actually relatively well understood. Nicotine, as you know, is the psychoactive component in tobacco, and about 80% of it is converted to the non-active cotinine, and the conversions are catalyzed by the liver cytochrome P450 enzymes, primarily the CYP2A6, but a little bit the CYP2B6 as well. And CYP2A6 is involved in the metabolism also of a variety of xenobiotics, several pharmaceuticals, several carcinogens, and a number of coumarin-type alkaloids. And we know that these genes are polymorphic in the population, such that some individuals have increased activity of the enzymes for which the genes are encoding, and some have reduced activity of the catabolic activity of these enzymes. We also know that cotinine is conjugated with glucuronides for excretion, and these are conjugated by the UGT family. These two are polymorphic in the population with varying abilities of increased and decreased activities to conjugate cotinine. So that we need to study variants in these genes if we want to understand something about inter-individual differences in nicotine metabolism. But in addition, the most robust findings to date in this side of the chart relate to the nicotine acetylcholinergic receptor activity. We know that nicotine directly enhances cognitive functioning such as attention, memory, and learning via stimulation of these receptors. But in addition, we know that excitation of these receptors provokes firing of subcortical dopamine secreting neurons that are part of the mesocortolimbic dopamine pathway. Dopamine is known as the reward pathway. It signals pleasurable experience, and it is critical for the reinforcement efforts that promote self-administration of nicotine, and it's the reason why people are unable to quit smoking easily, because smoking results in all these pleasurable feelings. And we also know that nicotine augments dopamine release via opioid pathways, glutamatergic pathways, and GABA pathways as well. So any variance in any of these genes can alter the level of nicotine, can alter the response to nicotine neurotransmission function, and can explain why certain individuals are more likely to smoke heavily, and are less likely to quit. And I'm going to give you a very few examples. Here we have two smokers, both of whom maintain the identical nicotine levels in their plasma. Both of them smoke cigarettes containing the same amount of nicotine. However, this individual is only smoking 13 cigarettes a day. This individual is smoking 23 cigarettes a day. The only difference between these two individuals is in their CYP2A6 genotype. This individual is a wild type, and this individual has one wild type and one null allele, and this individual therefore has a slower metabolic activity of the CYP2A6. There is less conversion of CYP2A6 to cotinine, and therefore smokes fewer cigarettes per day to maintain nicotine levels. This individual, we would presuppose, would have a far easier time to quit than this individual. And we can use such information, hopefully, to develop novel pharmaceutical interventions to promote smoking cessation. And as the slide illustrates, very important, nicotine-dependent individuals adjust their smoking behavior to maintain their nicotine levels. There have also been Candid gene studies looking at the cholinergic nicotinic receptor region as a risk factor for a variety of different nicotine-dependent phenotypes. This one is looking at age of smoking and showed that individuals with certain variants started smoking earlier than others. That is an example of a Candid gene study. And here is our Candid gene study looking at CYP2A6 genotype and the number of cigarettes smoked per day. Here, these are individuals who had slow-acting genotype. These had the normal, and the people with the normal functioning CYP2A6 alleles smoked on average over 5 cigarettes more a day than the slow-acting, the reduced activity genotype carriers. Here we took, this is a variant in one of the genes that is associated, that it controls the nicotinic acetylcholinergic receptor function. There is a marginal increase, but it's not statistically significant in individuals with the risk alleles. However, when you combine the two, the CYP2A6 risk alleles and the chromosome 15, which is where this genotype is for the nicotinic acetylcholinergic receptor genotype, when you combine these two, you can see that individuals who smoke the most have both adverse genotypes. So this is a nice example of a gene-gene interaction. And we see the same type of approach when we look at the Fagerstrom index, which, as you know, is a score which measures the level of nicotine dependence. It's based on a variety of measures such as number of cigarettes smoked, the time to first cigarette in the morning, and so on. And here you can see again individuals with both adverse genotypes had the highest nicotine dependence levels, and this trend was statistically significant. What is the implication of such research? Well, clearly such inter-individual differences in nicotine metabolism can alter the risk for becoming nicotine dependent and even for starting cigarettes. Certainly it can affect the amount consumed and the ability to quit. And what can we do about this? If we could find inhibitors of the enzymes or drugs that slowed the activity of the enzymes, these would provide novel approaches to treat the dependence. It could certainly reduce the amount smoked gradually and therefore help people to stop smoking. And we could even perhaps use these approaches to prevent people, young adults, from even starting to smoke. And there are subgroups of young adults or adolescents actually who are at higher risk for trying cigarettes and becoming dependent smokers. So it could be used both for treatment and for prevention. And this is the goal of smoking cessation, not to treat individuals as one size fits all. There have also been pathway and genome-wide association studies targeting these receptor genes, which I have mentioned several times. And let me give you an example. This is the chromosome 15 locus, which has 3 different nicotinic receptors on it. And you can see here there have been a variety of studies with the phenotype here being heavy smoking compared to light smoking. And almost every study, except maybe 1 or 2, 3 actually, have been associated with significantly increased risk of heavy smoking. And you can see here we had over 14,000 heavy smokers, 10,000 light smokers. The risk was 1.33 for heavy smoking if you had the adverse allele, and clearly this was very statistically significant because of the large numbers of cases and controls. And this is the type of meta-analyses that are very important to replicate the findings from genome-wide studies. But the story does not end there. Clearly we now know that the nicotinic receptors have an impact on nicotine dependence and smoking intensity. But as I've said, the story doesn't end there. Subsequently, 3 genome-wide association studies of lung cancer were performed, and this is the one from our group, and we showed here a strongly significant signal on chromosome 15. And it turns out that this locus is the locus for cholinergic receptor alpha 3 and 5 receptors, and 2 other studies found the exact same risk estimate, and a subsequent study in 2009 also confirmed it. Now, we know that bronchial epithelial cells selectively express these subunits. We know that the affinity of nicotine for the receptor is actually greater than the affinity for the for acetylcholine, which is the physiologic agonist for these receptors. We know also that NNK, which is the tobacco-specific carcinogenic nitrosamine, also interacts with these receptors, and by doing so it causes a cascade signaling of a variety of growth factors, including VEGF. It activates the AKT pathway, it activates and apoptotic pathways, all of which are hallmarks of cancer development. So we feel that there is a direct lung cancer carcinogenic effect as well, and it's the same chromosome 15 locus that I showed you before for smoking intensity. And here is the forest plot for lung cancer, and here we see also all studies showed a significant association with this. This is one of the genes in the chromosome 15 locus. It's so important it's called Mr. Big, after a certain series on television. The summary odds ratio was 1.31, and it was strongly statistically significant. We also did another meta-analysis using our International Lung Cancer Consortium. We had 17 studies in white, 6 studies in Asians, and 1 study in African Americans, and we were able to replicate the finding again for Caucasians and African Americans, but this particular the SNPs that we studied were not significant in Asians, although other loci have been identified for Asians. We noticed that there was no association in never smokers, but there was clearly an association with the number of cigarettes smoked per day. These findings would suggest that the association with those SNPs are largely due to their effect on smoking dependence and smoking intensity. However, we did find higher risks for those SNPs in younger patients, and in theory younger patients are lighter smokers than older patients, and you would expect the reverse if the effect was only due to smoking intensity. And here is some of the data from our own study. Along here we have high, medium, and light smokers, and here is the risk genotype, and this is the wild type. And you can see here that the risk is highest in the light smokers, intermediate in the medium smokers, and lowest in the high smokers. And this would suggest to us, therefore, that the genetic locus in this region on chromosome 15 influences both susceptibility to lung cancer and smoking behavior. 2 other SNPs of interest turned out in the lung meta-analysis. This was our favorite, chromosome 15. This is chromosome 6 with an apoptotic gene as well as a mismatch repair gene, and this is the telomerase gene, which has been implicated in several other cancers as well. So these were the 3 top hits in all the GWAS that have been studied to date. And we looked at the 2, the one from chromosome 5 and the chromosome 15, and we showed a gene dosage effect both for former smokers and for current smokers, that individuals who had 4 or more risk alleles had the highest risk. So this is called a gene dosage effect, and it was a very interesting finding. However, the next and most important question we have to ask, what is the clinical utility of doing these genetic association studies and of constructing genetic profiles? Well, here we have a risk prediction model for lung cancer. The blue one is the basic epidemiologic model, which includes age, gender, smoking variables, family history of lung cancer, history of chronic obstructive pulmonary disease. And the green one is the same model, adding the 3 top SNPs. Well, if you can barely see the difference between the 2 curves, that is my point, which is that these very small increases in discriminative accuracy of risk models is unlikely to be of any diagnostic or predictive utility. So we have to go back and do a lot more homework before we can say that adding genes to any risk prediction model will improve the performance of this model. So what are our next steps in trying to deal with this question of the biological basis of lung cancer susceptibility? It is quite clear that we are going to identify many more common variants that contribute to lung cancer and any other type of cancer. However, they are likely to have too small effect sizes to reach significance in genome-wide screens. And we've come to the stage when there are probably diminishing returns in predicting disease risk from common markers, and it probably seems sensible to invest a greater effort in functional investigations of all these biological hypotheses being turned up by GWAS. So what do we need to do? Well, of course, we need to take the loci and causal variants that we find and do replication studies. It's critically to do those, and these need to be done in certainly external populations in different ethnicities as well. And once we need also to take the regions of linkage disequilibrium and the replicated SNPs, and we need to do fine mapping studies, resequencing study, and epigenetic studies of these regions, irrespective of annotation or the relationship of the loci to nearby genes. And then the next step is to do extensive functional genomic studies, and these could be both in vitro, such as expression studies with constructs of nearby genes, as well as in vivo studies such as mouse knockouts of nearby genes or fish or cell knockdowns of nearby genes. And finally, we can do mammalian quantitative trait loci of the synthetic regions. And these, of course, epidemiologists require to work with systems biologists, bioinformaticians, and so on to do these studies, and certainly with biologists as well. Another approach is to look at the genetic determinants of intermediate phenotypes, and for lung cancer this could be looking at genetic determinants of DNA repair capacity, which we have shown is a risk factor for lung cancer. Individuals who smoking causes substantial DNA damage, and you require competent nucleotide excision repair systems to repair the damage. We've already spoken about smoking behavior. We know that chronic obstructive pulmonary disease is a significant risk factor for lung cancer, and it's also obviously caused by smoking. We could look at genetic determinants of COPD as well. And finally, we could look at extreme phenotypes, and for lung cancer, maybe we could look at high-risk probands from extensive families with lung cancer. We could look at early onset. We could look at light smokers and so on. And we need in our risk assessment models to study much more carefully gene-gene and gene-environment interactions. And finally, as I've said before, the gap between discovery and clinical translation is very wide. Our long-term goal is to develop personalized prevention and therapy approaches. We would like to predict response or toxicity. We would like to help identify molecular targets for developing therapies, and certainly we need to improve our risk prediction so that we know how to apply prevention programs in the population. And I hope I've convinced you that an integrative multidisciplinary approach is key. And I think the U19 approach that our NCI has developed epitomizes this type of team science multidisciplinary approach which can address these issues. Now the next question is, what mechanisms link energy balance and obesity to cancer? We now know that adipose tissue is an active and major endocrine and metabolic organ, important not only in the regulation of energy balance, but also lipid metabolism through the release of a variety of peptide hormones. And we know that there's intensive studies trying to explore a variety of these intertwined pathways such as cytokines, adipokinins, sex hormones, insulin, IGF pathway, the mTOR signaling pathway, and so on. I would argue that epidemiologic studies per se are unlikely to accelerate mechanistic understanding, and we need to think outside the box and to look at a variety of different options, such as the genetics of obesity, the role of activated brown fats. We need to answer the question, is the association reversible if obesity is treated? And then this links to the final question, and that's the role of the microbiome in obesity. So this is not a political map, rather it's to show the severe epidemic of obesity that we're now facing, with the red representing more than 55% of the states having adults who are overweight. It's a serious problem. There are 30 million new cases of obesity a year. We know that obesity is associated with the risk of a variety of cancers, including colon, postmenopausal breast, endometrial, kidney, adenocarcinoma of the esophagus, and even hepatocellular carcinoma. The attributable risk for endometrial cancer from obesity is about 50%. For colon, it's about 15%. Even as important, we now know that obese patients have poorer survival, and those who gain weight, weight gain is a predictor of poor survival, particularly in prostate and breast cancer. It's been estimated that the healthcare costs by 2030 will reach over 900 billion cases a year. And of course, what we could do about preventing 300,000 deaths a year if we were able to address and treat and prevent the obesity epidemic. We know that there is a genetic control to obesity as well, and to risk or susceptibility to this obesogenic environment. It's estimated that the heritability component for obesity varies between 40 and 70%. It's difficult to arrive at a precise estimate. We also know there's something called the thrifty gene hypothesis, which was proposed in 1962 by James Neal, and this was to explain why certain ethnic groups tend toward obesity and diabetes. And this hypothesis postulates that certain genes in humans have evolved to maximize metabolic efficiency in populations subject to times of food scarcity, and therefore in times of abundance, such as now in America, carriers of these genes which promote metabolic efficiency predispose them to diseases caused by excess nutritional intake, such as obesity. We are also recognizing that there is an exceptionally complex control of feelings of hunger and satiety which is controlled in the arcuate nucleus of the hypothalamus through a variety of neurohormonal pathways with integration of different cortical signals, and as well as a response to external signals in the environment as well. And this homeostatic mechanism is very delicately balanced, and the balance can easily be upset by a variety of genetic and other factors as well. The first robust obesity gene was called the fat mass and obesity associated gene, shortened to FTO, and it was shown that a variant in exon 1 of FTO was associated with BMI, basal metabolic BMI. It was actually, that's body mass index. It was actually, the study was designed to look at risk of type 2 diabetes, but they found that this effect was entirely mediated through BMI, and so it's only indirectly associated with diabetes through the effect on BMI. The risk allele of this FTO gene is common. It's in about 70% of Caucasian and African American populations, and between 20 to 40% in Asian populations. Homozygote carriers weigh on average 3 kilograms more than wild-type carriers, and they have a 1.5-fold increased risk of obesity. The risk allele is thought to increase food intake and decrease feelings of satiety. It's been replicated in many child and adult studies and in different ethnic groups. What is interesting about this association is that the effect of the risk allele is attenuated by about 27% in carriers who are physically active. The mechanisms of action of this gene are under intensive study. It's thought that it's associated with decreased lipolytic activity of adipocytes, and it is thought that it demethylases single-strand DNA, but this is a really ongoing area of research. And sad to say, it only accounts for about 1% of total heritability of body mass index, so we've got a long way to go. And in fact, A variety of other GWAS have been conducted for obesity-associated genes. Here is our FTO, and this is the Wnt-catenin signaling pathway SNP, which has been associated with inhibition of adipolysis. And this is the melanocortin 4 receptor pathway, which is also associated with early onset monogenic obesity. So what have we learned from GWAS? And this was published in 2011. It's probably out of date by now. About 58 loci have been shown to contribute to polygenic obesity, which is the common type of obesity. And of these, 36 SNPs have been robustly associated with BMI. 4 genes only were also involved in monogenic obesity, which is actually not a bad number. However, as with the smoking, combining risk SNPs has no clinical utility in risk prediction as of yet. I also wanted to just spend a few minutes talking about physical activity, because we now know that research into physical activity has lagged considerably behind research on other modifiable risk factors. We also know that there are considerable individual differences in response to exercise, and that there's a substantial familial aggregation component as well. We know that physical activity is obviously associated with adiposity. We know that adiposity is associated, white adipose tissue is a source of pro-inflammatory cytokines and causes systemic inflammation, and this causes a variety of different diseases, including not only a variety of cancers as we've shown, but other diseases as well. And if you look at the association in a meta-analysis between physical activity and colorectal cancer, you can see that in both prospective and retrospective studies, there is a significant association, a protective association between physical activity and colorectal cancer risk. We also know that there are, as I've said, substantial familial aggregation component. It is quite well documented that the propensity to be a high or low responder aggregates in families and is influenced therefore by genetic factors. And this is a slide which shows the gains in maximal oxygen consumption, which is this VO2 max, between pairs of twins. And here you can see there's a substantial commonality, a good correlation in the twins, the interplay resemblance in response of identical twins, and this is much greater than it is within twins than it is between twins. So there is definitely a genetic component in response to physical activity. We also now know that there are 2 different types of adipose tissue. This is white adipose tissue, which, as we know, stores energy. It has large vacuoles, and it is a source of a variety of pro-inflammatory cytokines, as well as a variety of other peptides and hormones. And this is brown adipose tissue, which looks different. It's brown because it's a large number of mitochondria, it has small vacuoles, it's very well supplied with blood vessels, and it's richly innervated with sympathetic nerves. And unlike white fat that stores energy, brown fat expends energy. It's present in newborns, where it's said to represent up to 5% of body mass, but it involutes in early childhood very rapidly, and it's thought to help infants who can't shiver by producing what's called non-shivering thermogenesis, or heat. However, we now know through a variety of non-invasive scanning that adults do have some brown adipose tissue as well. And if you look here, here is a PET scan with a FDG tracer to detect metabolic activity, and you can see here the PET scan shows uptake of the tracer, and these are areas of metabolic activity in the supraclavicular region. Here we have the CAT scan, which identifies these areas as fat. So if you superimpose the two, here these are areas of brown adipose tissue. So as a result of imaging like this, we now know that brown adipose tissue is more likely detectable in women than in men, and in leaner individuals than in fat individuals. We also know, out of interest, in transgenic mice that have decreased brown adipose tissue are obese. So the very interesting findings. Now here is another interesting finding. These are images from the neck and upper thoracic region of a healthy individual who was put in a cold environment and a warm environment, and these are transaxial, coronal and sagittal images. And here you can see increased activity and uptake of the tracer in a cold environment in all three views, and here we see nothing. So this suggests that individuals in cold climates would be likely to have more heat production from brown adipose tissue. So we are left with the questions: does the lack of brown adipose tissue lead to obesity, or does obesity lead to lack of brown adipose tissue? Is there an obesity drug that we can find that would affect energy expenditure and not appetite? Because we know those haven't worked so well. There's been an observation that people on beta blockers have less active brown fat. So the question is, could we find a beta-3 selective agonist that would be effective to promote more brown fat and to be an anti-obesity agent in humans? If we're failing all that, we could perhaps put everybody in frosty spas to upregulate their brown adipose tissue. And in fact, a very recent publication in Nature identified a newly described myohormone called irisin after the Greek goddess, and this has been found to convert white adipose tissue cells into brown fat-like cells with their same mechanisms of action. This irisin is induced with exercise, as shown here both in mice and in humans, and they showed that with the increase in level of irisin, there was increased energy expenditure, there was slight weight loss, and there was reduced insulin resistance with an improvement of metabolic syndrome. So the question is, could this be one of the long-awaited exercise drugs? I think the authors were naturally and wisely very cautious. They said whether long-term treatments with irisin or higher doses would cause more weight loss remains to be determined. A very important question is cancer risk associated with obesity reversible? If we could show that it's reversible, this would be a powerful behavioral incentive. I've just taken two studies to demonstrate this. Until now, we've not been able to ask this question because we did not have large cohorts of individuals who lost weight and maintained the weight loss. But now we do, because about 200,000 bariatric surgeries are being performed every year in the United States. And these are two studies that followed up bariatric surgery patients and compared them to obese patients who did not have bariatric surgery. And they found in both studies, with a median follow-up of about 11 and 12.5 years respectively, that there were significant reductions in incidence of all cancers, mainly in women, and also mainly obesity-related cancers. And in terms of, they also found significant reductions in mortality for all cancer types. Now we know that bariatric surgery has profound effects on the metabolism, and in fact it's called metabolic surgery. And as you can see here, it causes a reduction in sex hormones due to decreased conversion from androgenic precursors by the aromatase enzyme in adipose tissue. It causes a reduction in lipid peroxidation and oxidative stress. It reduces levels of pro-inflammatory cytokines. It decreases free fatty acids, and most of all, it ameliorates insulin resistance, makes the tissues more sensitive to insulin, and reduces IGF-1 levels. And in fact, there's been a tremendous effect in reducing obese subjects with type 2 diabetes, reducing their needs for oral anti-diabetic therapy. However, we don't know which of these metabolic effects is responsible for the reduction in cancer incidence, and we therefore need to do very extensive systems biology approaches to try and sort this out. And we can also use in vivo studies with animal models to see which pathways are most perturbed by these types of effects. And as leads to my last question, we know that metabolic surgery has a profound effect on the gut pH and therefore on bacterial flora in the gut. So the final question is, what are the relationships between the human microbiome and obesity? We know that the gut microbiota are the largest collection of microbiota in the body, responsible for about 2 pounds in weight, and it would fill the surface area of a tennis court. They have an extremely important role both in physiology and pathophysiology. We know that the microbiome is influenced by the genotype of the host, by the presence of inflammation, which increases permeability of the membranes of the gut and allows pathogenic bacteria to infiltrate the gut. Individuals on vegetarian diets, probiotic foods, supplements, certain pharmaceuticals such as antibiotics and proton pump inhibitors, and even the climate can affect where the food is grown. All of these have a profound effect on the microbiome, and the microbiome in turn has an effect on the host. In terms of cancer risk, which I'm not going to go into, we know that it does actually expose the host to N-nitroso-carcinogenic compounds produced by the bacterial metabolic processes. We know that there can be chronic immune stimulation and tolerance, which leads in fact to atopic diseases, inflammatory bowel disease with risk for cancer, and gastric cancer as well. In addition, it's thought that the microbiome can have an effect on metabolism of endogenous and exogenous hormones as well. And there could in fact be a systemic effect on cancer risk. But I'm only going to talk here about the microbiome and obesity, and we've discussed all these host factors which can impinge on the distribution of the gut microbiome. It's important to recognize that the gut microbiota have an extremely important physiologic effect. They are responsible for breaking down indigestible polysaccharides such as fiber to short-chain fatty acids and monosaccharides, which provide an extra 10% of daily energy intake in adults. These short-chain fatty acids, as I said, are converted into triglycerides and into glucose in the liver. And in addition, this stimulates lipogenesis, leading to obesity. And we now believe that there is such a thing as an obese microbiome, which has an increased capacity to harvest energy from diet. And I'll give you an example in a minute why I say that. But in addition, there is the bidirectional neurohormonal gut-brain axis, which sends some information to the hypothalamus about the energy status of the host and can promote meal termination or hunger feelings and increasing food intake. And this is an extremely important part of the control of hunger and response to hunger. But we also know that the microbiome can actually impact this gut-brain axis. And it's been shown that germ-free mice display alterations in stress responsivity, and in fact there is a reduction in anxiety in germ-free mice compared to conventional mice when they're exposed to stress situations. So this can also impact the very fine bidirectional balance of feelings of satiety and fullness and hunger which goes to the hypothalamus. And we know that there is an association from both preclinical and human evidence. We know genetically obese mice have different gut microbiota than their lean siblings in terms of the type of predominant phyla within the gut microbiota. We also know that transplanting the cecal gut microbiota from a normal to a germ-free mice increases their body fat with no increased intake of food. And we also actually know that germ-free mice have to consume about 30% more calories than normal mice to maintain their body weight. We also know that obese mice, I said there's a change in the distribution of phyla, and they have fewer gram-negative bacteroides division phyla than their normal counterparts, and these are more effective at releasing calories from digested food, so they have what we call an obese microbiome, which generates more energy from food that's eaten. We also have human evidence. 12 unrelated subjects were put either on a carbohydrate-restricted or a fat-restricted diet, and we showed that as they lost weight, there was a progressive increase in the bacteroides phylum compared with the other major components. So there was an increase, and here this shows that this was correlated with the change in body weight, irrespective of whether they had a carbohydrate-reduced or fat-reduced diet. So it's clear that there are changes in the that you can manipulate the gut microbiota and decrease energy production from the diet. And certainly this could be drastic, but it could be an approach to obesity treatment. There are a variety of other unanswered questions which I won't have time to go through, but I'll simply list them here. Is there such a thing as a core microbiome? And how stable and resilient is an individual's microbiota throughout one day or during his or her lifespan? What causes diversity of the microbiota? How similar are microbiomes between members of a family or community or across communities in different environments? What affects the genetic diversity of the microbiome, and how does this diversity affect adaptation to different lifestyles and to various physiological or pathophysiological states? We don't know that either. And what is the effect of antibiotics on environment health? And how do we study this complex interplay of the microbiome, host genetics, diet, obesity, and a variety of other adverse effects? And it's clear that in order to answer these questions, we need very well characterized cohorts with serial but comprehensive biospecimens for metabolite profiling. And again, we need multidisciplinary teams and technologies to characterize the microbiota in multiple organ sites and to see the association with disease, with cancer risk, with obesity, and a variety of other physiological and pathophysiological states. So the next steps in addressing obesity, diet, physical activity, the microbiome, and cancer risk, we need to do much better at measuring phenotypes. And in terms of BMI, there are a variety of new ways to measure anthropometry with scanners and sophisticated ultrasound machines. We should certainly focus on intermediate phenotypes with all the new omics platforms that are available. And in terms of eating patterns, we can instead of just recording what they've eaten, maybe we could get them to do, with their cell phones, to take pictures with digital photography. Clearly, as I said, for the lung cancer studies, we need to design smarter studies with extreme phenotypes. For example, there's a South Asian population with a very unique distribution of body fat. We could study cohorts of very lean individuals. There are some. We could capitalize on existing cohorts such as bariatric cancer patients, or the children's health study, which is enrolling the 100 representative children from before birth to age 21, and this would be a wonderful time to evaluate the interrelationships between diets, nutritional status, microbiomes, and the immune system through metabolic profiling and studying the microbiota. And the same approaches that I mentioned for the post-GWAS would apply to the study of obesity as well. And we could also take genetically controlled mouse models. We could give them typical Western diet or physical inactivity, and we could then see, using a systems genetics approach, what molecular mechanisms are perturbed. And again, this needs to be a multidisciplinary integrated team. And some people have advised that we should use the same type of health warnings that have been so successful with tobacco. And certainly we can have public health policies such as the mayor of New York has already instituted, with a ban on 16-ounce sodas being sold. But we also have to consider the issue of personal responsibility. We all believe that in the old days epidemiology was lost in translation. For example, we knew about lemon juice protecting against scurvy in the 1600s. It took another 260 odd years before the British Board of Trade demanded that citrus be used for all its smokers, same thing for all its sailors, same thing with smoking. The first case control studies were published in 1950. It took 14 years for the Surgeon General report. The same thing with obesity. But my question is now, is slow translation over? In 2007, Science magazine said GWAS were the breakthrough of the year. In 2008, Time magazine said the DNA retail test was the best invention of the year. We had now the era of direct-to-consumer marketing of genetic tests, and we have expectation that genomic information will become integral in personalized healthcare delivery, and there are new terms floating around such as public health genomics and personal genomics, and people are expecting magic. However, we're now entering a period of disenchantment. There was a paper published in the New York Times a decade later, genetic map yields few new cures. So I think we need to take a sane approach to all this and take the message of Hunter and Kraft in 2009. What advice should we offer about genetic testing? The prudent answer, they said, is not now, ask again in a few years. And I'll just add, seeing it was written 3 years ago, a few more years. And finally, I would like to quote from the late Dean of the School of Public Health at the University of Texas in Houston: "If it ain't fun, it ain't epidemiology." Thank you.